Welcome back for more AP Biology. This particular lecture we're going to deal with phylogeny and cladistics. So the question that we're going to attempt to figure out is, how can we justify the fact that there are evolutionary relationships between organisms? So when we look at evolution, we can divide it into two categories. We have what we call microevolution and macroevolution. Microevolution is going to deal with how populations are capable of changing, whereas macroevolution deals with how we end up forming brand new species. So they're the same thing, just on different scales. One is small and isolated, one of them is much, much bigger. When we look at organisms, we like to put them into categories. So among them would be like saying, oh yeah, you are such and such species. But what do we mean by that? Yeah, we can give a definition of same organism or same population, same location, same time, reproducing. They have viable offspring that also reproduce. But what else could we mean by the term species? And that is a term that we use in classification. Classification is a way of putting organisms into boxes. The most nitpicky of those boxes we call a species. I can clump like species together to form what we would call a genus. I could clump like genera together to form a family, like families to create an order, like orders to make classes, like classes to make phyla, or a phylum, like phyla to form a kingdom, like kingdoms can form a domain, and like domains we just call life. And we've dealt with this for a very long time. So like we could say, oh, you belong to Kingdom Monera or Kingdom Animalia, or you are part of phylum, well, arthropods or arthropoda. So when we do that, because, well, these are all things that happen to have an exoskeleton and they're kind of crunchy and they turn out to have lots of limbs on them, well, that would make sense. Let's stick them all together. The catch is, if I start really examining these things, what you would expect is how I classify things based on how they look should also match evolution. They should also match genetic information. They should match protein information. And it turns out, even with a simple example like the arthropods, this actually doesn't work. I can classify them based upon how they look, but if I start talking about their evolutionary past and how I can try and link them together, this doesn't work so great anymore. So we need to have a new way of classifying. And that new technique of classification is what we call cladistics or phylogeny, which is, let's put you into those categories. We're not getting rid of the categories, but we're going to clump you in based upon your evolutionary past. So we can have all of these different types of organisms, but I need to be able to have a way to link them together. I need to have evidence outside of what they show. The drawings that we produce are called cladograms, and cladograms will tell us a few things. First thing is, they tell us points of commonality, what we can call a common ancestor, and that's where we get these little branch points. So each of these branch points, which are labeled as 315 million years ago, or 148 million years ago, or 166 million years ago, each of those branches is a way of saying we had a speciation event going on here. The length of these lines, either vertical or horizontal or radial, whatever, the length of the line, what that tells us is how long did it take between these events. Meaning, how long did it take to go from this branch point to get to that branch point. The length of time is reflected in the length of the lines. What I should be able to find when I look at cladograms is groupings of similar organisms. So as I look at this one here, what's telling me is the monotremes are less related to the other mammals as the marsupials and the eutherials, things with the placenta, turn out to be. The marsupials and the eutherials are more closely related than would be the monotremes to either of those. And the reason for that is we have a common branch point. So what do we use when we make these things? Once upon a time ago, what we would do is we'd look for commonalities, kind of like what we would do when we dealt with basic classification. So I could look for, well, do you have an amniotic egg? Do you happen to have holes in your skull? Do you happen to have live births? Do you have a placenta? Do you have hair? Do you have feathers? Do you not have feathers? Do you have limbs? Do you not have limbs? How do you regulate your body temperature? And I could start to put together one of these cladograms. Each of these reasons to cause a branch is what we call an outgrouping. Meaning, if I were to have an outgrouping of amniotic egg, I can say you have an amniotic egg or you don't. And from this list here, amphibians don't have an amniotic egg, so they are excluded, they are outgrouped based upon the amniotic egg. If I then look for openings in the skull for muscles to attach, what we notice is, of all the remaining organisms, turtles don't have that. Therefore, they're off to the side. Everything else past it contains those traits. If I look at hair, well, some organisms do, some organisms don't. So I can now separate from there. If I look for, do you happen to have limbs or not, we can separate from there. I can look at, do you regulate your body temperature? Yes or no. Do you have feathers for scales, or do you just have scales? Okay, yes or no. Do you have live births? Yes or no. Do you happen to have a placenta? Yes or no. And I'm just making a series of yes-no questions. Everything beyond it, the answer is yes, it has it. Everything to the behind, the answer is no. Turns out when we start putting these things together, we can make different groupings. So I can have a grouping that all has a common origin. So if I look at this branch point here, A, B, C, and D all share the same branch point. So we could call that a monophyletic group. They all come from the same origin. I could have a grouping like C, D, E, and F that actually has separate branch points because C, D, E, F are kind of all split apart all over the place. And we're not including everything there because we're excluding A and B. So we're picking and choosing from our branch points. We call that paraphyletic, meaning the groups are near each other. And then I can kind of start to mix and match from all over the place, and that we call a polyphyletic group. When I look at the arthropods, they turn out to be polyphyletic. They actually don't follow evolutionary patterns. We're picking and choosing from different locations. Nowadays, what we can do is we can test cladograms as hypotheses, meaning I can say, I propose this is the best arrangement of these organisms. These are where the branch points are. I can justify each of these. How did I come up with this? Well, I can do it based upon observable traits. I can do it off of the environments that they live in. I can go off of molecular information, DNA sequences, RNA sequences, protein sequences, metabolic pathways, what have you. So when I look at these, which is let's organize animals, what I can see is I have one option on the right, A, and I have another option on the right, B. The question is, which one makes more sense? So if I look on the one on the right, I'm going after how things look. But if I go after how things work molecularly, which have nothing to do with how you look, well, they do, but to a lesser degree, I get the picture on the right. The question is, which one is correct? And what we always have to think about is the term parsimony. Which one is the easiest answer? Or, more appropriately, which one is the easiest to explain? Which one has the fewest changes in it? And if I follow that train of thought, what you would realize is option B makes more sense than option A does. Option A, when you look at it, okay, we're clumping things that look alike together, but the problem with that is we have some things that we can't quite explain. Meaning, I have one explanation, yes, these are how these look, but now I have to add in additional explanations to say these are why these organisms are grouped together. Whereas in option B, I don't have as many things that I need to explain. I don't need to tell you what I mean as often. So I'll give you a simple example from all this. So if you start looking through, what you can wonder is, well, tell me about the organisms that swim. And if you look down here on A, basically our swimming organisms are all clumped together. So here we happen to have a swimming organism. There's another swimming organism. Yay, look, all the swimming organisms should be next to each other because they all swim. If I start looking at other sets of data outside of they swim, what you realize is, oh, well, shoot, I actually need to split them apart. And we just got two separate events, but molecularly this makes more sense, which means parsimony is going to win. If I start taking into account all the different mutations that need to take place in order to make an arrangement, I can derive some statistics. What is the likelihood of getting all these mutations to cause this outcome? And what we need to do is decrease the probability, meaning let's make it so it's highly likely that this option will work and these options here are less likely, which is a nice way of saying let's follow parsimony. So when we do this now, we can start with our original idea, which is let's look at basic characteristics. But what we do now is we start with that as the basis, and now we start adding in gene sequencing. So we can look at one gene, see what pattern we get. Now let's look at another gene. What pattern do we get? Now let's take another gene. Let's see what pattern we get. Let's take another gene. Let's see what pattern we get. If you do that enough with hundreds, if not thousands of genes, you can end up generating a whole bunch of tables. The catch is, if I look at fossils, I don't always get that option. So when we deal with fossils, all we can really go off of is anatomical structure. It's just what we happen to have to play with. So we're putting these things together. We're looking at fossils from the past. We're taking what we have now. What we'll notice is sometimes we have pieces that are missing, like we can't connect all of the dots. We call those missing links. And it's among the things that people like to attack about macroevolution, so speciation, is, well, you can't explain what the organism is in here. So the response usually becomes, well, I think I know how to connect the dots. So we start hunting for where we think the dots should be, and every once in a while we will find more connections. The problem with that is it then begs, oh, well, now you have even more missing links, which means, okay, you do more hunting and we start to fill in, which means you'll then say, oh, but where are the missing links for those organisms? And the problem becomes missing links create more missing links. And at some point it becomes I found 95 of the 100 in a row of the missing links or the transitional species. I'm pretty sure my pattern is working. If I had 3 of the 100, this could be an issue, meaning to get from point A to point B, we could imagine there's 100 points in between, and I have 3 of those potential 100 points. Yeah, that might not be a good argument. But if you have 95 of those 100 points in between, yeah, your model is working. For a lot of organisms, we have the 95 of the 100. We don't have all of them because not everything turns into a fossil, because it turns out fossils are hard to make. And when I look at fossils, when anyone looks at fossils, what we notice is if we put them into phylogenetic groups, I can't speak today, what I find is if I plot those over time, we have periods of lots of them, and then they disappear. Lots of them, then they disappear. Lots of them, holy crap, they disappear. Lots of them, they disappear. Lots of them, they disappear. Those periods where we end up having wipeouts, we call an extinction event. In Earth's history, we've had 5 major extinctions. Of these, most of them you've never heard of, like the Ordovician and the Devonian and the Triassic you've never heard of. You might have heard of the Permian extinction, which is the one that wiped out like 99% of everything that existed at the time. The most famous one of them all is the end of the Cretaceous, which is the let's wipe out the dinosaurs. The catch with these things is they open up brand new ecological niches, which means we could have more evolution. And if I start to get big wipeouts, I have more space for things to grow. Well, the catch is, as of right now, A lot of scientists are claiming, this is from July of 2017, we're in the sixth mass extinction. We are living through it. The other events happen because of the climate. Things are going on with the climate, as far as we can tell. This one here, we're thinking it might have something to do with the climate. The difference is we are aware that we can manipulate the climate. So this one might be on us.